we're excited to thank you. Welcome to everyone on Zoom as well as here. Today's event is Postmortem Human Brain 101. Okay, yes. So welcome and thank you again for your patience. Today's speaker is Dr. Sabina Translational Neuroscience Research Lab. The medial thalamus, the entorhinal cortex, and the olfactory systems. Dr. Berretta began her research training at the University of Bologna in Italy. Then she moved to the lab of Anne Graybiel at MIT. She was studying circuits linking the motor cortex to the basal ganglia. She also worked in the lab of Francine Venez at McLean, where she was developing an animal model for studying GABAergic abnormalities in schizophrenia. And yeah, thank you so much for joining us, and we'll get started.
Am I good with the microphone? Okay. Well, thank you so much for inviting me, Parisat. I am really pleased to be here, and thank you so much for everybody to taking the time to attend this seminar, both in person and on Zoom. Thank you for all the questions that have already been submitted. I'm gonna try answering some of them during my talk, but please feel free to answer questions. There will be a couple of breaks where we could ask questions, or maybe leave them at the end if they would take longer to discuss them. So as Parisan mentioned, I work at McLean Hospital, and as you can see from the title of my talk, which I couldn't resist translating into Latin, the mortui vivos docent, is the old saying that the anatomist would say to justify using cadaver to understand human anatomy. And what we are doing here is, in a way, that focusing on the brain. And what I would like to talk about today is not only how that can be done and give you some examples from my lab, but also tell you how it is made possible by collecting brain donations. So at McLean Hospital, let me see if I can figure it out to the, right, I sort of wear two hats. One is the our research lab, which is the Translational Neuroscience lab, and the other is working with the Harvard Brain Tissue Resource Center. So just to give you a brief outline of what we will discuss today, I'll tell you a little bit about what is the role of human postmortem investigations in the context of studying both the human brain, but also brain disorders. How is this made possible? So the role of the Harvard Brain Tissue Resource Center is one of the many tissue repositories available. So we'll talk a little bit briefly about how does a brain donation work, but also from the point of view of investigators, how to request tissue and what kind of information may be available. And I'm happy to talk more at length about that later on after my talk. And then I'd like to give you just a few examples of work, taking examples from work that we have done in the lab. The work I'm going to present is all unpublished, but it's kind of more fun to discuss it that way. So what is the role of human postmortem studies? The main point is that brain disorders are particularly complex and difficult to understand, in part because they are complex our brain is, or the brain in general across species. And that is compounded by the fact that the very many brain disorders are a complex, does it type going a little bit? Yeah. Okay. The complexity of the brain in general is compounded by the fact that many brain disorders, in fact, a large majority, are a complex interaction between genetic vulnerabilities and environmental factors that can happen at different times in life, from prenatal life all the way to older age, and therefore they're particularly complex to study. So that is why we really basically need to use all the tools that we have in our box to study these brain disorders. We need genetic, for the reason I said, a lot of these brain disorders, in fact the largest majority, have at least a genetic component, some more fast than others. We can use in vitro studies, so trying to mimic the cellular pathology and ask a question about how different cells may be altered or mimic certain abnormalities that we are expecting. We certainly need animal models. They give us a unique ability to manipulate circuits, to look at behavior, to look at cell by cell, and dynamically understand how abnormalities in the brain may interact with each other. But obviously, animal models need something to model, and that cannot be just the behavior, because it's very difficult to mimic the behavior or the symptoms, rather, of brain disorders within an animal model. So we need clinical studies, we need to understand patients better in many ways, their behavior, their symptoms, how they respond to medication, how they respond to certain treatments. We obviously need human brain imaging. That is a really powerful method that allows us to look at the brain while it responds to stimuli, to tasks, how different brain regions are activated together. You can assess people that you are working with clinically. I would say the human postmortem brain is almost complementary to human imaging studies. On the one hand, we have disadvantages. We don't see the patients doing live for the majority of the time. We obviously cannot do dynamic studies, although I'll tell you a little bit toward the end of the talk on how we are trying to address this. But we see molecule by molecule, cell by cell in every brain region. So we have a lot more resolution than imaging does. And in general, I think we would all agree that there is no method that is perfect and unbiased. We need all of them and try to kind of combine them in a way that we can answer questions and understand what the limitations and the advantages of these methods are. So what kind of things can we do with using human postmortem brain? Many of you are already working or have been working for a long time on human postmortem. The oldest approach, perhaps, other than looking at the anatomy, is perhaps histology. Told us a huge amount in terms of pathology of very specific disorders. We can use immunocytochemistry or histochemical techniques to look at specific type of neurons, their morphology, their rotation within certain brain regions, the cytoarchitectonic of different parts of the brain. We can certainly do subcellular studies. It's difficult in the human brain, but it certainly can be done. So we can, with different microscopy techniques, we can look at things like dendritic spines, for instance, as shown here, or even ultrastructural level studies, such as here shown as a migraine graph. We can look at proteins in different ways. The most classical way other than immunocytochemistry, could be Western blots. Still very useful methods. I would say that methods are as valid as the question that you're asking, and if you're using the right method, then it doesn't matter how old it is. But there are very novel and continuously improving ways of use the brain proteomics and looking at post-translational modifications, trying to bring it down to the cell level. And in fact, something that is important to consider is that the amazing step forward that we have taken as neuroscience and the tools, methodological tools that we have doing perhaps the last 10, 15 years have brought to a really intense, anyway, interest in looking at human postmortem brain, because we can ask the questions that we couldn't have even dreamed of asking even 10 years ago. We can use RNA-scope, as you might be familiar. It's basically a new library of what we used to do as inside hybridization, but now we can do it in multiplex. We can look at literally molecule by molecule inside different types of cells. The new methods for single cell sequencing, so single cell RNA-seq, for instance, have brought us an amazing opportunity that is expanding every day to look at the single cell level of gene expression and characterizing cells according to cell states, something that we didn't used to be able to do. We used to think in a more rigid way about cell types, but that is changing very rapidly. And that is also, for instance, with tools for spatial transcriptomics that is really bringing us very, very fast, faster than we thought, about being able to ask the questions that, again, we wouldn't have been able to even dream about when I started this work, for instance. So all this work, and these are just examples, by no means this list is exhaustive, but I just wanted to give you a flavor of different things that can be done. But all this is made possible by the process of brain donation. So I'm gonna switch gear for a little bit. I'm gonna try to go as fast as I can on this part, but please feel free to ask me questions if you have urgent ones anytime, please, but also at the end of this talk. So I am the director of the Harvard Brain Tissue Resource Center, one of the oldest and perhaps largest brain repositories in the country. It was founded in 1978 by Dr. Bird, who was an expert in Huntington disease studies, and I became director in 2014. A little bit about what is our mission as part of the Brain Bank now is to make research on brain disorders and the human brain possible. We actually know about the human brain a lot less than we think we do. And this is because for a long time we have relied on work on other species. And although that is perfectly valid, there are obvious species-specific differences. Our brain is not better or worse than any other species. It just has species-specific adaptations. And therefore, not everything that we know from rodents or even non-human primates is true, particularly now that we can go, again, at the exquisite level of detail. So studying the human brain, understanding how it is similar and different from other species, but most importantly, try to understand what are the pathological underpinnings of brain disorders. And we do this by collecting brain tissue specimens from across the country. I'll tell you a little bit about how that works, and then redistributing them out to investigators across the world. An important mission of this work, in addition to, or as a result of research on brain disorder, is to combat the stigma that is associated with brain disorders, pretty much all of them, but particularly psychiatric disorders. The reason for that is because we tend to think about particularly psychiatric disorders a sort of behavioral disorders, not coming from, not arising from changes in the brain. That is obviously a misconception. There is no change in our behavior that is not coming from what our brain is doing, but leads to stigma, because if it is not a biological change, then we assume that person could change it, could do better, could snap out of it if they are too depressed to get out of the bed. If we understand what are the biological underpinnings of those symptoms, then we could treat this disorder like any other disorder. Nobody would be blamed for being diabetic. Why are we blaming people for having major depression or schizophrenia? Or are we considering them less than other people? So that is a particularly important aspect of the work we do, and one that we try to pay attention. Very briefly, our brain repository, the Harvard Brain Tissue Resource Center, is one of 6 repositories that are part of an organization run directly by NIH called the NeuroBioBank. And here you have the 6 different repositories. This was started in 2013 and brought to a major advance in how brain recoveries and brain tissue distribution is done, because we work together, we harmonized the protocol. Investigators can request tissue to the NeuroBioBank, which means that 6 different repositories will be able to contribute to their tissue request. And we are fortunate to collaborate with many different foundations with whom we do outreach and on whose behalf we collect brain samples. And this is particularly important for relatively rare disorders. You can see here many that are, barely known, such as CDKL5 deficiencies. This is for Christensen syndrome, Tourette, and many, many others. ROHAD is a particularly rare metabolic disorder that also involves brain changes, but also for other disorders that seem less rare, it is particularly important for us to put it out there how important it is to contribute to this by considering to become a brain donor. And I'll shamelessly hit on this point several times during this talk. So a little bit about ourselves. We collected, actually, I should update that, over 10,000 brains since we have been operating. However, of course, we distribute tissue in a way faster than we can acquire it. So we currently have about 4,000 brains that we can draw from for distribution. People can register, and I'll tell you a little bit about it. And we are organized in a way that we can collect brain donation as a fresh specimen within 24 hours. This is thanks to a very dedicated, passionate team of people at the brain bank that is on call pretty much all the time. Will pick up the phone or be at the lab dissecting a brain, no matter what time of the night or day it is. And then, as I mentioned, we distribute across the country. This is an outdated slide, but it's in 5 years, the number of brains that we had collected across the country and the number of specimens we sent out. Just to give you an example of the breadth of this work. So as I mentioned, anybody can register to become a brain donor. That is not a consent, but it's a way people use to have a conversation with their families or to discuss brain donation, to make sure that the family is aware of the wishes, or sometimes it is the family that register people if they're not able to do it themselves. So how does it work? Very briefly, timing is of the essence. So in order for brain specimen to be suitable for research, we try to keep what we call the postmortem time interval. So the time from when the donor passes to the time we are done with dissection and the tissue is stored within 24 hours. And this is whether the donor was here in Massachusetts, in the middle of the country on the West Coast. It needs to be done within 24 hours. So I'll tell you a little bit about how it is done, but something that we rely on and we collaborate with is the family or somebody on their behalf needs to call us as quickly as possible, either when the donor may be imminent or within the first few hours. And that, as you can imagine, is something very difficult and very challenging to ask the family to call us as soon as possible within a few hours of losing a loved one. We can talk a little bit. I know that there were some questions, and they are very interesting questions on how it is done, how do we work it out. So very briefly, this is Darren Chernicki, one of our clinical liaisons. People can call the brain bank anytime. There will always be somebody on the other side of the phone answering, no matter what day or time it is. We first need to screen. There are a few exclusionary criteria that we need to go by. Once we are able to accept the case, we will do an informed consent with the legal next of kin in a secure manner remotely. So we actually never meet the family, unfortunately. We have a network of recovery specialists, oftentimes pathologists, across the country that will go and recover the brain for us. So we don't do it directly. We wouldn't be able to within the time frame. We have couriers that will bring it to our laboratories at McLean Hospital, either driving it or putting it on a flight, depending on where the donor was. And I won't take too much time, but it is important to understand that sometimes, despite our best effort, it doesn't work. There are several reasons. The brain may not be suitable for research. We have very few exclusionary criteria. Obviously, for instance, penetrating injuries to the head would not be compatible. There would be too much damage. We are very careful to rule out certain infectious diseases. We're particularly worried about prion disease because, as you can imagine, it would pose a risk not only to the pathologist doing the recovery, to us, but also to investigator receiving the tissue. So that is the most stringent of the criteria we have for recovering brains. We don't knowingly accept donors that are HIV, Hep B, and Hep C positive. Sometimes the family doesn't know, so we do a serology on all donors. But and then there may be some logistic questions. We're not called quickly enough. We're not able to recover it for some reason. But we do our best to not to let this happen. So overall, we accepted the largest majority of the brain donations that are offered to us. When the brain comes in, we have a team waiting in our laboratory. They're there at 3 o'clock at night on a Saturday. For some reason, there is always a brain coming at midnight on New Year's Eve. There will be dissectionists there ready to work on it, no matter what time it is. And again, it is essential because time and temperature are actually the most important factors in making sure that these specimens are suitable for research. And then we store the tissue. Part of the brain, half of the brain is dissected and frozen. Part of it is fixed and goes through neuropathology and is also available to investigators. After that, we work with the family to recover as much information as we can about the donor. This is because the time is so limited when at intake that we need to go with what the family knows. So if the family tells us the donor had dementia, that's what we know and that's what we take it for. But then obviously we need to gather as much information as possible in order to characterize the brain and provide information to investigators. So we'll work with the family to recover health records. We'll send out a packet with the questionnaire, release of health records. And then, as I will show you, we do other tests. One of them is neuropathology. So the fixed part of the brain will be dissected by a neuropathologist. This is Derek Oakley, our neuropathologist at the Brain Bank. And the neuropathology report not only goes into the overall information collected by the donor, but is also shared with the family. And that is often something that the family really appreciates, in part because it's some sort of closure, but also because at times the diagnosis in life wasn't so clear. And having the neuropathology helps them understand what their loved one suffered from, and in some cases, what risk there may be to our other family members, depending on the disorder. Once we have collected all this information, so health records, a family questionnaire, the neuropathology, and so on, we do a consensus diagnosis to establish which different diagnoses at the clinical level, neuropathology, systemic, and genetic, the donor had. We collected sort of a history of the donor, and then at that point, the tissue is ready for distribution. So tissue can be requested, and I'll tell you a little bit more later. I'm happy to answer questions, of course, that may be something of interest. But tissue from our repository, as well as the others part of the NeuroBioBank, can be requested directly from the NIH portal. And this is, again, a really important advantage to investigators. It used to be that different brain banks operated somewhat independently. So an investigator may have had to either stick with the tissue repository, who may not have had all they needed, or ask different repositories, and protocols were not standardized. Now this is well coordinated. We work together. Investigators can choose samples from any repository. We will work the tissue request together. And then, of course, the final point that is really to do research. So we distribute tissue to investigators across the world. There are no, although tissue requests are vetted very carefully, there is no particular exclusion. So any investigator anywhere in the world, whether in academia and in the private sector, can request tissue from us. Excuse me for a second. So a little bit more, and again, I'm happy to say more. We do a number of quality control measures. We measure pH, serology, toxicology. We collect as much demographic information, clinical history, family history. We assess medication and substance use in many different ways, both from the health records and the family questionnaire, but also from toxicology. And again, we do a consensus diagnosis. Sorry. It's asthma. Are there questions in the meantime? Of course.
Very interesting. I'm very curious about your brain. But first, so what the questions that I have, one is the health information, if I understood correctly, it's respected that.
Correct.
Oh, okay. And for the neuropathology, exactly what aspects?
So it's a standard neuropathology report, so it will depend on the brain disorder. Okay. So we take what we know already from the family. Oftentimes we already got some of the health records. The neuropathologist will do an assessment, first of all, the overall brain, but based on what disorder, for instance, dementias versus Huntington versus Parkinson, they will do different tests.
So it's the first they're like a few settled. I feel bad for asking you questions.
No, please do. You give me time to recover. It happens to me all the time. Yes, if
there's someone is healthy, then is there a set of pathology assessments that you do?
Yes, because the point of the the neuropathology is not only to confirm the diagnosis, if it can be confirmed with the neuropathology, but also to assess whether there was something else. So in healthy controls, in people with psychiatric disorders, in which there is not that sort of first level layer of pathology, we need to understand what else there was. Interestingly, even for brain disorder that have a classic first layer neuropathology, it doesn't always match the origin of the clinical diagnosis. So we get a lot of cases in which the clinical diagnosis and the neuropathologists actually don't talk to each other, which is actually really interesting.
And then the last question, you mentioned that anyone can request tissue, but I'm curious to understand how you prioritize the request and what's the timeline of when I was request.
Yeah, those are. Anybody has a tissue, I would appreciate.
I'll have one.
Thank you so much. So anybody can, as I mentioned, ask for request tissue. There is several layers of vetting. So first by NIH and then by the tissue repositories. So NIH will look at things like, does the investigator have funding to do that, to carry out the study? If it's okay if the investigator does not have prior experience using human postmortem, but then we will ask, for instance, to do a pilot study to see if the work that they propose to do is possible, if they have the expertise, or whether they need a collaboration that will help them with them. We also look at things like, do they have a power analysis? Are they requesting too little, too much? Do they have a strong rationale? But we do it not in the way, you would review a grant submission to NIH. Is more from stewardship of the tissue. So it's more doing our best to honor the trust that was put in us and the intent of the donor and their family in donating their brain. So we look at things, in a grant application, the more subject you put in, the better it is. We look at whether you really need 300 subjects, or could you do the study? Would it be powered well or sufficiently with 200? But anything that, so when investigators put in a request, what we advise is, look carefully at your design, ask for what you need. That will always be distributed out. The delay in distributing the tissue depends a little bit on a number of things. A very large request will take a long time. Sometimes the time is actually longer for tissue requests that are a bit more complicated, so that we are not sure whether they know how to do that particular assay. We might ask for a pilot that will delay. But again, the only purpose of this is to use the tissue in the best way possible. Oh, yes, I'm sorry. Yes. So the questions were about how tissue requests are vetted, how they are prioritized, how it is decided to who will get the tissue. And again, to request the tissue, please use this site. However, I'm always happy, myself or other people in my team, to answer questions. So if you look at the site and you are not sure how to handle it and how to put together your request, we're always happy to work with you. I'm sorry. So the question is whether we can collect the data before the donor passes. We, at the moment, collect relatively limited data when they register. But if they're not registered, no, we can take a call without the donor being registered. We are gearing up to collect more. It's complicated because this becomes human subject studies. So we already have very stringent IT security and confidentiality protection, but that would kind of amplify it. But it would be a really important way. And I should say that everything that I said, it relates to our tissue repositories. Many different brain banks work in different ways. So for instance, there are a lot of brain banks that follow people longitudinally and then eventually may collect a brain donation. So this is not how a brain bank in general works. This is how this particular brain bank works. But that's a great question. So a little bit about some examples, and I'm drawing from work in our lab. So a lot more can be done with this tissue, of course. So one first study that I would like to tell you about, and overall, our lab is really focused on emotion regulation. So brain regions and circuits that are involved in emotion and how they are affected in psychiatric disorders, but also in many other disorders. That's an important component of this. We tend to split psychiatry from neurology. In reality, psychiatric symptoms occur in many other disorders that we typically think of as neurological. So for instance, dementias typically have important psychiatric symptoms. So does Parkinson's, so does Huntington, so do many other. So in reality, what we are more and more interested in our lab is to look at symptoms transdiagnostically. So depression, for instance, is very common, not only obviously in major depression or in bipolar by definition, but also in many, many, many other brain disorders that we tend to classify in different ways. So the question is, what underlies depression? And are the mechanisms the same in dementias versus major depression, for instance? So this first example, and again, this is just, I'm going to go very briefly. I can't really get into detail, so you might have to take my word for it, but I'm happy to chat later on. One of our main grants is a Conti Center, is focused on anxiety, and particularly on two peptides that we know are involved. One is corticotropic releasing hormone or factor, depending on whether you study rodents or human. So CRH in depression and anxiety. And one particular small component of this study, so it's a Conti Center, 5 different projects, all based on McLean, working on anxiety disorder, headed by Bill Carlson and Kerry Ressler. And in this study, together with Kerry Ressler, I don't know why that is there, but we looked at the expression of CRH, so corticotropic releasing hormone, is a peptide that has for a long time known to be involved in anxiety. But what we wanted to ask was, if we take 2 different cohorts, in addition to healthy control, major depression, and PTSD, both have a strong component of anxiety, we see differences between these particular disorders, but we were also looking at a very specific area in the brain, which is the central nucleus of the amygdala. So one particular brain region that is known to sort of implement anxiety responses, and we can talk more later. But as you can see, the first layer of result we got was a bit perplexing, because we did see an increase in PTSD with respect to major depression, So you could argue, okay, major depression has less anxiety, that may be why it is different, but we didn't see a change in control. And as we dug a little deeper and tried to understand what were the factors that were determining this change, we realized that actually it wasn't necessarily PTSD in itself, but that the majority of the factor that most accounted for this was actually suicide. So once we took out the healthy controls, put together major depression and PTSD, and then compare the donors that had died by suicide versus those that didn't, we realized that in fact, it was the death by suicide that was accounting for the increase in CRH in the central nucleus of the amygdala. We also looked at exposure to trauma or stress, and particularly comparing exposure in early life, so childhood in this case, versus adulthood. And this is because exposure to stress and trauma is well known to have an effect on both depression and anxiety as a vulnerability factor. And to make a very long story short, indeed what we found that the effect that we originally detected was across the two diagnoses, a combined effect of death by suicide and early exposure to trauma, so childhood trauma. We didn't see the same effect, for instance, in adult trauma and suicide. You could argue there is a little bit there, but not as strong of an effect as we find in early trauma and suicide. So although this is consistent with our hypothesis, it was really surprising to see such a strong effect, and particularly speaking to that idea of looking at disorders or looking at symptoms transdiagnostically to try to understand what are the components that may be underlying the symptoms. The other study that I would like to tell you about is a study that we are doing in collaboration with Steve McCarroll and one of his very many talented postdoctoral fellow, McLean. So in this study, we looked at almost 200 cases between healthy control and people affected by schizophrenia within the dorsolateral prefrontal cortex, so Brodmann area 46 in this case. And again, I'm just going to give you a snippet of what we found. We found a really interesting sort of gene expression program that is coordinated between neurons and astrocytes. And specifically what we found is that in both glutamatergic neurons and GABAergic neurons, the gene expression investment of these neurons in synaptic components is correlated with the cholesterol biosynthesis in astrocytes. And this is important because neurons can make their own cholesterol, which is essential for building new synapses and maintaining them. But in reality, the majority of the cholesterol that is used by neurons for synaptic maintenance and synaptogenesis is provided by astrocytes. So astrocytes make the cholesterol, synthesize it, and then transfer it to neurons to be used in for synaptic functions. So the two programs, synaptic components in neurons and cholesterol synthesis in astrocytes, make sense. We all have it. They're correlated in our brain. However, what we found is that there is a significant decrease in schizophrenic. Steve and his team called this program SNAP, which stands for Synaptic Neuron Astrocyte Program. It makes it easier to describe and use. And again, what we found is that SNAP, as this sort of coordinated program between neurons and astrocytes, is very significantly affected, in fact, decreased in schizophrenia. this is important because there has been a long sort of investment in understanding synaptopathology in psychiatric disorders, but this tells us that it is really a contribution of two different cell types at the very least that impacted. And in fact, a lot of the component of SNAP is an astrocytic component and is related to risk factors for schizophrenia. Now, we were saying at the very beginning that one of the drawbacks of postmortem is that we can look at the changes that are in the brain of people that have those disorders, but we can't really do dynamic studies. We cannot manipulate the system to ask questions. So that's why we need all the other tools that we were discussing and many more that I didn't list. But there might be a way that our lab is working on in which we can use postmortem tissue in that way. So, for instance, in the study that I just described, how would we understand the interactions if we can't manipulate the system? And something that our lab has been doing lately is find methods to extract live glial cells from human postmortem brain. So these are fresh preparations. So before we do anything with them, before we freeze them or fix them, but we are able to extract, and this is very new work, it's not published. We are at the very beginning, but it's exciting enough that I wanted to tell you about it. So we are able to extract both astrocytes and microglia. So these are examples, for instance, of astrocytes in culture. And we were just doing quality controls. These cells express GFAP and S100 beta, showing that they express markers that are specific for astrocyte. We do the same thing for microglia. And this is the work, I should say, by really talented people in our lab. Natalia Kolosowska, who is a postdoc and brought in this method to the lab, and then Anna Lalli and Elizabeth Lawton that are working with Natalia, and they're really playing a major role in developing these protocols. And one last slide on this is, this means we can do functional studies. So in this study, for instance, we were using a phagocytosis assay to test whether, in this case, microglia extracted from human postmortem brain. And you can see at the moment, we tend to focus on the white matter, so just because it seems to be easier, but we are also looking at gray matter in different brain regions. We wanted to test the phagocytosis as an effect of exposure to immune factors, in this case, interferon gamma and LPS. And as we postulated that there were the phagocytosis, which is a sort of a functional kind of housekeeping function. So it goes down when the cell is challenged because it needs to be engaged in other function, as we expected was decreased. So as a way of a brief summary, and I know we're a little bit over, I try to convince you that human postmortem studies do provide a unique way to get insight into the molecular and cellular underpinnings of brain disorders, that recent very powerful methods, particularly at the single cell level, many of them developed here in Harvard Medical School, as well as many other groups, of course, are offering an unprecedented opportunity to study these brain disorders and also to understand the human brain better. And I gave you a few examples of findings in our lab that use human postmortem in different manners. My last two slides are going back to brain donation and sort of making one more plug for brain donation. We can all help with this. And this is because we need healthy controls as much as we need people with brain disorders. In fact, healthy controls in a way are the most difficult group but they also go in every single study we ever do. So because of that, and because we need a comparison group both for health study and for group comparisons, everybody can consider brain donation. And just to give you a sense of how each brain really contributes to this, each brain, because we only distribute as much as an investigator needs, goes into studies, in hundreds of studies, both within the United States and across the world, and therefore it has a huge impact. As last and most importantly, my thanks always and my gratitude goes to all brain donors, families of brain donors that went through such a difficult choice at a particularly challenging and painful time in their life. And of course, NIH as part of the NeuroBioBank, as well as NIH funding to my lab, of course, and you for your kind attention. Thank you.
Thank you very much for your talk, very interesting and informative. I have a couple of questions. One of them, how did you decide and how many slices you divide the brain?
That's a good question. So actually protocols have changed with time, and particularly as our work we post. So sorry. The question is, how do we decide on how many sections we divide the brain? It's dictated by different considerations over time. This has been standardized not only by the time our tissue repository became part of the NeuroBioBank, but it changed relatively recently because we're also one of the, we as NeuroBioBank, one of the main sites contributing tissue to the Brain Initiative program, so the BICAN, the sort of the cell census within the brain. So at the moment we cut coronal sections that are about 5 millimeters thick, and we leave them intact. And 5 millimeters is about as thin as you can go on a fresh tissue. In fact, we had to figure out tricks to make sure the tissue doesn't collapse. But it allows you to find even small brain regions, which in thicker section end up kind of at times trapped into the thickness of the section. So we used to do it according to anatomical landmarks. Now we cut evenly at 5 millimeters. That works quite well because again, you see even small like the subthalamic nucleus in the section where it is.
My second question is regarding to that. If some researcher can get the brain tissue, but he is interested in a specific area,
Yes.
And that area is small.
Yes.
How do you decide how many slices you can give them?
So we won't give the slices. We will redissect a coronal section to get the particular brain region that an investigator will need. And we work with the investigator to understand how much tissue they need. So if you go, for instance, on the NeuroBioBank site, you will see that there are guidelines that sort of try to help you understand that. So for instance, depending, I don't know, if you want to do single cell RNA-seq, typically 100 milligram is more than enough for several runs. But if you're doing something that requires sectioning, you might need more or less. So it's decided as a collaboration with investigators, and sometimes it takes a little bit of negotiations, but it works out.
And my last question is about the controls.
Yes.
When you or the researcher requests the brain tissue, you have to do to the control brain? Or
yes, so the way it works with the NeuroBioBank, you actually choose first the cases that you would like, and then the tissue preparation and the brain region. So you will, depending if you're doing a group comparison study, you will choose your controls so that they are matched to the group you're trying to study. So you literally pick case by case, brain region by brain region. Then it will be vetted by the NeuroBioBank and by each tissue repository to make sure that you're getting what you really need, that you're getting your study is well powered and all that.
Thank you.
Yeah, thank you. There was a question that was submitted ahead of time that is particularly close to my heart and with a lot of things that we're doing at the moment, which is in part what motivates families to, for instance, go ahead with a brain donation, but also how do we think about the different cultures and how would we address that. And this latter one is particularly important and is something that we are trying to do quite a bit of work, and I would love to hear your thoughts. But to the first part, which is the easiest one, families are motivated by many different reasons. At times, it is the person itself that will become a donor that expressed the wish to become a brain donor. This happened many, many times, both for healthy controls for people that are unaffected, even more often for disorders that are very obviously impacting the brain. All of them do, but some it is more obvious for people. Sometimes it is the family that is motivated for the same reason. A brain donation is really a legacy. You are helping others with the same condition or with many different conditions by helping research. And that is what really motivates most people to donate and motivates the families to donate. And I can give you many, many examples of families that really went out of their way to either carry out the wish of their loved one or just do it. We had a few years ago, an elderly lady that knew her husband wanted to become a brain donor, lived in a relatively isolated area. We didn't have the technology we have now. She needed to use a fax to send us back the consent. At 2:00 at night, walked a mile to the next gas station to use their fax so she could give us the consent. This is not unusual, but it's most of the motivation. In other cases, as I was mentioning, is people will want to understand what was the condition that was affecting the loved one, and sometimes it's less obvious than others. The other question, which I will only touch upon briefly, it would require a long conversation, maybe we can have it if anybody's staying later on, is as in the majority of research studies, there is a tendency of having mostly white as participants. There are very obvious reasons for that. I don't need to list them. I'm sure you can figure them out. Lack of access to healthcare and restrictions and inequalities are probably a major component. If people don't have access to proper healthcare, why would they want to help research and medical care? Long history of abuses and atrocities committed in the name of research on vulnerable communities and minorities are obviously an important factor, but there are a lot of other components that intermingle and intersect with this that we may not be able to understand completely. So what we are trying to do in collaboration with other people at the McLean Hospital and with other communities is to really study this, understand what are the reasons, whether they are cultural, social, religious, and they might be different, obviously, for different communities that impact this. At the moment, I talked about how brain donation is important to research, but the majority of donors in our tissue repository are white, 98%. So the research that is done is not completely relevant to everybody in the world, and certainly does not reflect the composition of the population in our planet. So something that we're trying to do, and I welcome any suggestion where, is not necessarily go out and ask for brain donations, but understand what are the reasons, how we can generate trust, how can we work with different communities to fix this, because it is a bias that at the moment is important and not addressed as well as it should be.
We can do one last question. We have one on Zoom from George, who says, thank you for the talk and amazing efforts. From what I understand, there's an increased frequency of psychiatric brain disorders occurring or being really reported in youth, particularly teens and adolescents. Does the Tissue Center have many adolescent donations? Are these much rarer and more difficult to find in adult donations?
Absolutely. I mean, for many reasons. One is, thankfully, younger people die less often, although obviously suicide is a huge problem. It is also a problem that, as I was saying earlier, psychiatric disorders are often less understood in our general culture as disorders really impacting the brain in a way that can be studied. So there is less awareness of why these brain donations may be important. Oftentimes, unfortunately, death at young age or death by suicide, this takes the family by surprise. They may not be ready to think about a brain donation. So we try our best to represent all ages, but it is obviously difficult and another big challenge we all have.
We have to wrap things up. Thank you so much for your wonderful talk. If anyone's staying for the lunch, please here.
Thank you.